We have now transformed E. coli cells with the DNA plasmid that contains all the components required to produce RFP, and give them at least 48 hours to grow at 37 degrees. Let's explore what our expected ideal results look like and what some less than ideal results may look like. Let us begin by looking at the P+ plates. As a reminder, P-ARR is the plasmid that we are transforming into the P+ E. coli cells, complete with everything required to produce large amounts of red fluorescent protein when arabidose is present in the environment. For P+ cells that grow on ABE medium, there's no antibiotic selection pressure and there's no arabidose to promote any plasmid protein growth. Therefore, we expect to see a lawn of E. coli bacteria growing as fast as the ABE agar nutrient medium allows them to grow in the petri dish. Some of these cells have the engineered plasmids, some won't. However, with no selection pressure, the presence or absence of the plasmid doesn't really prohibit growth. For P+ cells grown on ABE amp medium, there is now selection pressure in the form of the antibiotic in the growth medium. As such, only the cells that have been transformed and thus have the ampicillin resistance gene to allow them to grow in the presence of the antibiotic will grow. Depending on how dense and well-transformed your original competent cell culture is, you may get a lawn of E. coli cells, or a less dense growth of distinct individual colonies. For P+ cells grown on ABE amp era medium, you should see similar numbers of colonies as you saw on the ABE amp plate, since all of the same cells should have ampicillin resistance. However, most of the colonies should also be pink, or in the process of turning pink as a result of RFP production. There may be a very small number of colonies that don't turn pink, whether it's due to a break in the plasmid DNA that cannot be fixed, was incorrectly fixed, or something happened to the P-BAT or RFP region of the plasmid that prohibits RFP growth. For the P- cells that were not transformed with plasmid, they should only grow as a lawn on the ABE plate. Introducing any antibiotic into the growth medium, such as in both the ABE amp and ABE amp era plates, would kill any cell growth. If there's any growth on the ABE amp plate with P- cells, questions should definitely be asked. Now let's see what the results should look like on real plates. Most sites will grow the P- and P+ cells on the same plate, splitting down the middle of the plate for better comparison, and to cut down on reagent costs. As you can see from a recent transformation performed by the ABE LA site technician Karen Steinhauer, bacterial lawns grew on the ABE plate on both the P- and P+ side. For the ABE amp plate, only the P+ sides grew colonies. For the ABE amp era plate, the whole plate is used to grow P+ cells, and you can see an abundance of bright pink colonies that have ampicillin resistance and express RFP. I've also included the expected results from Lab 5 here from the Complete series, where students digest 2 different plasmids to generate 4 fragments, 2 of which are the fragments that make up P-ARR, the 807 base pair and the 4,495 base pair fragments. Students then perform ligations with this mixture to generate a plethora of different plasmids resulting from all possible combinations of these 4 fragments. Of course, only a small fraction actually produce the P-ARR plasmid. As such, you can see a mix of both white and pink colonies on the P+ plate on the bottom right-hand side, where the pink colonies are from bacteria that have taken up a P-ARR plasmid that was successfully produced from the ligation reaction, and the white colonies are from bacteria that have taken up a combination of fragments that include the ampicillin resistance gene, but not what's required for RFP production. However, we know mistakes can happen in class, as illustrated by the results from 2 student groups. In each picture, the top row shows the expected transformation results, and the bottom row shows an interesting non-expected result. All plates are, theoretically, from left to right, ABE, ABE amp, and ABE amp era. Take a look at these results and write down what you think happened to these groups. Remember, students are instructed to use the same cell spreader to plate the same type of cell, going in order of ABE, ABE amp, and ABE amp era. For example, they're supposed to use one spreader to first spread out P- cells on the P- half of the ABE plate. Then use the same spreader for the P- half of the ABE amp plate. They then do the same thing with P+ cells using a different spreader on the 3 remaining P+ sections. Alright, let's talk about the results on the left here. There seems to be some pink colonies on the ABE plate. But how? Remember, students use the same cell spreaders to go from plate to plate to plate, from ABE to ABE to ABE era. It looks like this group plated their P+ cells on the ABE plate first, then went straight to the ABE era plate, and then finally to the ABE plate. And some of the arabinose from the ABE era medium hitched a ride to the ABE plate. If you look closely, there's a gradient of pink colonies in the center of that cluster, fading to white colonies as the arabinose ran out as the cells were spread. On the right-hand side, we yet again see some pink in the ABE plate, but this time there is no pink colonies on the ABE era plate. This one is most likely a case of reversed plates. The ABE plate was mistaken for the ABE era plate, and vice versa. You should be prepared to assist your students in making sense of their results, which may differ than the expected results. Good teachable moments often come from this transformative lab.